My name is Su Miao, and I'll be talking about my research interests. Before I start talking about that, I would like to talk a little bit about my research background and experiences that I've had in academics and in industry. So I got my bachelor's and master's in biomedical engineering at Binghamton University. I worked in the bioelectronics and microsystems lab, which is in the electrical and computer engineering department. And my thesis was on stretchable and flexible microbial fuel cells, specifically working with the Pseudomonas aeruginosa bacteria cell. And I had started a collaboration with FIT, Fashion Institute of Technology, located in New York City as well during this time. It was a really good introduction into cell culture work, as well as designing experiments on my own and writing papers. Next, instead of going directly into a PhD program, I wanted more experience in wet lab work and working more independently, so I decided to apply for an industry job. The first job that I had was at Vigene Biosciences, located in Rockville, Maryland, where I was a research associate for AAV process development and manufacturing, and I also had experience in learning analytics on AAV. So during that time, I felt that I needed more science background rather than working on batch record reviews and manufacturing. So I decided to apply for jobs in Massachusetts. And there I got a position at TCR Squared Therapeutics, where I worked on lentiviral vectors instead of AAV in their process development group. And I worked closely with their T cell group, process development group, for transitioning between R&D and manufacturing so that we can transfer the protocol to a CMO. It was a short time that I spent there due to the amount of work that was given that wasn't what I was really interested in. So I had a friend who worked at Intellia Therapeutics who was starting her own lab, and I had the opportunity to join her there. Intellia Therapeutics was focused on CRISPR technology, utilizing AAV as one of the viral vectors to deliver the gene of interest to ex vivo T cells, and then also some in vivo work that involved AAV as well. There, I was a associate scientist working in the AAV process development and analytics. Specifically, I was more focused and experienced in the upstream work, and so it has led me to really become more interested in cell culture and upstream research. And that is when I came across other companies that were working on work besides CRISPR in the synthetic biology field, such as Ginkgo Bioworks. They're known as an organism company, which means that they tailor and customize their specific organisms that their clients are requesting, as well as other work that they're expanding into gene and cell therapy. And one of the works that have also been interesting that I've developed over the time that I worked in industry is a field called synthetic virology, which introduces the synthetic part of the research, but more implemented to find different types of capsids and design different ways of changing the virus into a more man-made synthetic virus. It's utilized for gene delivery or other therapeutic uses. And then other topics that I've came across are called cell-free systems, where they take the actual process that's inside of a cell, but then utilize it and harness it outside of the cell so that it's more easy to develop and produce rather than something that's more unpredictable as producing in the cell, which is what a lot of stuff in industry and academics is using right now with developing and producing AAV and lentiviral systems. So that's why it has brought me to my PhD that I'm doing at UMD College Park, and I'm really excited to rotate in different types of labs such as tissue engineering, which they also are using CRISPR for one of their projects that I'll be involved in, and then also a lab with AAV nanoparticle imaging and delivery, and then the third lab with focused on synthetic biology with the cell-free system. So